So what I'm going to do today is to give you a bit of an overview of some of the background behind why we put the project in place in the first place. Give you a bit of introduction to bioinformatics and some of our needs and some of the challenges that we see. I'm going to introduce Climb itself, and then I'm going to talk a little bit about what we've done and how we've done it. So in terms of bioinformatics and biological research, there's a long and rich background that sits behind the reason why we would want to do something like building a large infrastructure, and why, in fact, we would need a lot of computational resources in order to do the sort of analyses that we do. And it comes down to revolutions that we've seen in genome sequencing over the past sort of 20 or so years. And so for me, I'm a bacterial disease researcher, and for me, I guess a lot of it begins in 1995, when I was just starting senior school, when the first bacterial genome was whole genome sequenced. So this was Haemophilus influenzae. It was sequenced by Craig Venter, and it was published in 1995. In 1998, we had this system called MLST, which is a system for epidemiologically typing bacterial pathogens based on short sequences, and that came out then. And then in 2000, we had the draft of the human genome, and that was then finished in 2003. And this was all based on this technology called an ABI Sanger sequencer. And a Sanger sequencer could do 96 samples and would take about 3 days to do 96 samples. So genome institutes like the Wellcome Trust Sanger Institute, they would have a warehouse filled with these sequences running night and day, producing sequence that you could then stitch together to create a whole human genome. And that took a worldwide effort costing billions of dollars and taking 15 years to actually do. Then in the mid-2000s, we saw the release of these two technologies. So the first was Roche 454, which is now has now been retired by Roche, and the Selectra Illumina, which is still going strong. And then more recently, we've had these so-called third-generation technologies, PacBio, IronTorrent, and most recently MinIron. And what these technologies do is they make sequencing massively parallel. So they're high-throughput sequencing instruments. And what you do is you sequence millions or hundreds of millions of short sequences simultaneously on a single instrument. So what that means is, on your ABI sequences, you could do 96 reads a run, and that would take about 3 days. On the top-end Illumina instruments, you can do over 500 million reads per run, and that takes about 10 days to do. So we've gone from a situation where it took 15 years to sequence the whole human genome and the endeavour of research institutes all over the world to produce a single human genome, to the point where we can now produce multiple human genomes on a single Selectra Illumina instrument in less than 10 days. So the growth in life sciences, the growth in life sciences data, is really driven to a large extent by these technologies, which are easily bought, easily run, and will continuously produce huge quantities of data in a very short space of time. And what that means is that we can ask all sorts of interesting questions about variation within populations. So I look at how bacteria move around the world, how they change over time. But you can ask anything from questions looking at bacteria all the way up to questions about humans, about diseases like diabetes, or looking at cancer. And that's all done through the lens of whole genome sequencing. So we can routinely generate these huge quantities of data. Now, the problem that you get as a bioinformatician is, you come to the Cisco talk that preceded is really interesting, because you come to a university, and bioinformatics capacity in a university is the definition of shadow IT. Okay, it is the absolute definition of that. I hasten to add, this is not our land room, but it really sort of emphasises, I think, the potential when you get people who are not interested in setting a system up for a long term or designing a system to operate, within the strictures of actually think about things like reliability and maintainability, where your main task is to do research, and you have to get a system up and running that enables you to ask your research and answer your research questions. And so there's this real conflict there. And so when I arrived at Cardiff, I had the joy, and it was a joy, in many ways, of working out how was I going to do my research in this new place. I'm a bioinformatician, I work on genome data. I can get genome data generated quickly and cheaply by my collaborators. We can do it locally with sequencing instruments that we've got in Cardiff. But then how do I analyse that? How do I store that? And I was basically presented with, I got there and they were very, I asked them, do you have bioinformatics infrastructure? Do you have equipment for it? And they're like, yes, we have a server. And it's like, okay. And what about your storage? Oh yeah, we've got another server for storage. Okay, how are you backing up? It's raided. And immediately, you're starting to worry about that particular infrastructure, because it's there. There's a lot of people relying on it within groups within the university, and it's not really what you would think of as a robust environment where you'd want to be saving your data and doing your research. And within that, you then have this problem of, well, how do I get my scripts and software installed? So I worked at the Sanger before I moved to Cardiff, and I'd written a load of scripts there which were hard-coded to run on the Sanger high-throughput computing system, which runs LSF. Right? So it has LSF, it's got a nice Luster file system, so everything pretty much is hard-coded to work in that environment. And then I arrive at Cardiff, where first of all I don't have anything myself, but if I want to use the university HPC system, that runs PBS. And all the path names and conventions that you're used to on the Sanger system are now completely different. And so you've got to go through your 2 years worth of scripts and software that you've written to work on the Sanger system and recode all of it to work on a local HPC system. So how do you get your scripts and software installed, and how do you get them running? Where do you do it? Do you put it on the HPC system that has certain limitations and things they don't like you doing? Or do you just, you've got your startup package, do you just buy yourself a couple of servers and go that route? And the problem is that we've already heard, I think, about siloization that we see in universities. And when you're a new lecturer, when you come into a new place and you don't know anybody, and you've got, the edifice of IT services on one end, and you've got, an HPC group that are several buildings away on the other and don't respond to your emails and don't pick up the phone, probably for good reasons. At that point, you're very much reliant on yourself, and you've got to do something that works for you. And that's a real problem. And I don't think it's an unusual story, and it's a story that's going to get more and more frequent because, with the best will in the world, most of the PIs that you see in biology today don't understand informatics, don't understand, actually using the command line and using software packages that they've written to do analysis. And, so I guess my generation, people of my age and younger, are the ones who are going to be becoming the junior academics soon, and most of us are more literate and are able to do things perhaps that IT services don't want us to do. We're able to do it a lot better than the ones who were there before. So this is a growing problem. And so what we have is we have this paradigm, I guess, that Sanger talk about, which I've shamelessly stolen and sort of broadened out beyond sequencing. So genomics is one of several platforms that can produce large amounts of data very cheaply and very quickly. So we can now routinely produce extremely complex biological data sets in hardly any time at all, with very little thought, actually, about how we're going to handle that data and how we're going to manage it and how we're going to analyse it. And the difficulty is that while it's very easy to write the small costs or the modest costs for this into a grant application, the hard bits and the bits that you really need expertise for and really need knowledge for are the bits that sit below the surface that most academics are simply unaware of. And so our major costs and major difficulties don't lie with our generation of data. We can do that routinely as a service. It's not difficult. They lie with how we store it, how we share it, and how we analyse it. And so you've got this set of interlinked issues which are challenging no matter what way you want to look at it. And the added challenge here is that within a university HPC context, biologists don't do workflows that play nice on HPC. Okay, so I deal with large complex data sets, often using software which is not necessarily written to optimize things like RAM usage. So I'm a high RAM user, and I will analyze thousands of samples simultaneously in an embarrassingly parallel way. It would not be difficult for me to saturate a university HPC system with my workloads. And it's funny, when I talk to people at ARCA about the way they like to do their system, they like to have, they don't like to be 100% utilised, they like to have a bit of a wiggle room so people get their stuff going through quite quickly. At Sanger, the HPC system, high throughput system, is 100% utilised all of the time. And you know when you submit a job that you are going to have to wait for it to finish. Because the thing is that the system is saturated. And so it's incredibly well utilised. But that creates issues for HPC as well. So I mean, Sanger routinely has sort of upwards of 50,000 jobs in its job queue, which is starting to push the limits of the scheduler even. And within a university context as well, you probably don't want biologists coming wholesale onto your system to hammer it in that way. So we have these challenges in bioinformatics. We have challenges around the infrastructure, and we have challenges in terms of our storage and in terms of our high throughput computing capacity as distinct from our high performance computing capacity. A lot of our workloads are embarrassingly parallel. And we have this sort of this weird thing where we seem to either have embarrassingly parallel workloads or workloads which are almost single core or even single thread high RAM workloads. We have this really strange combination of workloads. We have issues around portability of our software. Very good at sharing software and our data. We've got lots of nice big databases that store the data and then cause problems when you try and download it onto a university system. And we've got lots of bioinformaticians who will have a GitHub page, and that's the standard way that we share our software. But that doesn't make the software portable. Just because it's available doesn't mean that it's very usable. And our data sets, well, you can put your genome sequences on the European Nucleotide Archive. We do that routinely. We do that when we publish our data, and quite often we'll do it as soon as we've got our data. And so it's publicly available. But actually, interacting with those databases is also highly non-trivial in how you can then get your data out in a way that you can reuse it, or combining data sets that have already been published. And then the last issue is that there are those of us who are pretty comfortable with accessing the command line, with writing software, with actually understanding the system. But there's a lot of biologists, and the vast majority of biologists, who really aren't. So there's an issue around researcher IT skills, and that affects the way that they act. So the idea of buying a server for a group, that's a very easy way for a wet lab-focused PI to understand a need. We have a need for some compute, and in the same way that if we had a need for doing PCR in the lab, we would buy a PCR instrument. And so you buy an instrument, a server, that does the compute for you. Okay, so it's a very simple link, a simple way of doing that. And so we have a lack of IT skills affecting that, but also then it affects what they can do. So I know lots of researchers who will flatly refuse to access the command line. Even if they've got big workloads to do, they will not access the command line. They don't have the time or the inclination to reskill in actually being able to do stuff over the command line, so an HPC system. And that's an insurmountable challenge, because as long as research councils are prepared to give researchers money to buy a server, there's no reason for them to move away from a graphical interface. And I don't think that's ever going to change, because the biologists are not trained to use computers. So we have an issue around IT skills as well. So I probably don't need to explain this, but I put it in anyway. So the idea being that if we have, we feel that the solution is actually a lot of those servers that people buy will sit around doing nothing for the majority of the time. So it's a really inefficient use of university resources. It's a really inefficient way of actually having hardware. Having single servers sitting on their own with connected into another server that has a RAID array doesn't give you any data security. And the simple truth is that if you want to do things properly, you have to do it at scale, and you have to do it with proper planning. And that's expensive if you try and do it on your own, but collectively it's actually quite cheap. And so the idea being that if we have a cloud approach, we can take this large shared infrastructure and we can share it out between our users. And virtual machines, I think containers will be a possibility, but at the moment we're working on virtual machines because it's conceptually easier. Virtual machines marry very well with the idea of servers. You can give a researcher a virtual machine. It's their own little playground. It's their own little silo. And it conforms to the idea that, we need to do something, and, we've got to do our analysis. And I ask for a server, and I get it in the form of a virtual machine. So conceptually, the idea of having your own space works well for biologists. It provides flexibility in terms of software that you can actually make available and what people want to do, which is great for great for what we want. And it actually meets the needs of a lot of researchers. And so we think that actually the cloud is a great model and is a way to actually overcome some of these issues. So I think, I probably don't need to sell this too much here, but the cloud is a mechanism for providing core infrastructure at scale. It's removing the need for local, and by local I mean within the department, for example, hardware maintenance and support. It's a way of presenting IT services out to a user community without having to say it's an IT service necessarily. You can actually make it potentially transparent to the user. And it's much more cost efficient. So it's much more cost effective potentially to research councils, but also to universities as well. And then because you've got virtual machines, that provides a ready-made mechanism for sharing data and sharing software. So it actually helps to kind of overcome this data reuse and software reuse problem. And finally, it also makes training easier. And the reason for that is if you do training, so when we do training around the country, they come in and they want to learn about pathogen genomics or whatever, and you teach people, whether they be everything from students all the way up to German professors of research, I've taught before. And they will learn how to do the analysis on the system that you teach them on. But then they go home and they find that there's a different system that they can't use. And obviously if you're teaching on a virtual machine that can be run on any infrastructure, whether it's a local infrastructure or something like Amazon, then immediately when they go home they can access the same thing they've been taught on, and so there isn't that jump that they have to overcome. And so that's a major advantage. So CLIME is a cloud infrastructure for microbial bioinformatics. It's an MRC-funded big data project. The total funds, I think, are about £8.4 million, and that's split between training and hardware provision and maintenance. And so it's what I call a public-private cloud. So it's public in the sense that it's not just for researchers at the 4 host institutions, which is Cardiff, Birmingham, Warwick and Swansea. It's public to the UK academic community, but it's private in the sense that it's available to the UK academic community rather than necessarily anyone who wants it. There are some exceptions to that, so industry and collaborators and people who are working with the UK community can also access. We have a set of standardised cloud images that implement key pipelines. So Public Health England, for example, have put their pipeline for bacteria analysis onto our system, so other people can now access that, including other government departments who couldn't access it within their local system. We've got a storage repository, so we can store data and images, we can make them available online, and the idea is that you can then access them anywhere. And so sort of moving towards this idea of an edgy roam almost for microbial genomics as a mechanism of accessing your data almost anywhere. And then obviously we'll provide interfacing to other databases from within the system. So we have very good collaborative links with the European Bioinformatics Institute, for example. And one of the things that we're working on is actually directly connecting into their nucleotide archive to provide that natively within the system. And we also have then the capacity within our infrastructure to support orphan databases and tools. So quite often there'll be useful databases or useful software packages which once the funding runs out, they're no longer hosted anywhere. And obviously if you can turn them into a virtual machine, you can carry on hosting them wherever you want at relatively low cost. So system outline, we've got 4 sites connected over Janet. We have different sizes of virtual machine available, like any cloud system. Able to support over 1,000 virtual machines simultaneously. That's an entirely arbitrary number, and it's actually limited at the moment by the number of IP addresses we've asked for from the universities. So we can support more within the infrastructure. But that sort of assumes a ratio of 1 CPU to 1 virtual CPU to 1 CPU and core, and 1 gigabyte of RAM to 1 gigabyte of RAM on the system. We have around about 8 petabytes of object storage spread across the 4 sites, and so depending on how we configure it, there could be anywhere between 2 and 5 petabytes of storage available with Erasure coding. We're currently working on exactly how we want to distribute that based on our respective use cases. We have 400 to 500 T of local high-performance storage per site. That's used for spinning up the virtual machines, and that's separate from our object store. And we're providing a single system with common login, and that's using, at the moment it's using Shibboleth, but we want to move it towards Moonshot. And then we've got between-site data replication. So we get our resilience not only by the fact that these systems are built to be resilient, but with high availability thought in on an individual site. In the university data centres, they've got things like, so in Cardiff they're in water-cooled racks with full power protection, with diesel backup generators, and the works. But we also have between-site data replication. So the data, so a user's data is present on more than one site. So if we lose a whole site, a user can log in and spin up a virtual machine somewhere else and still access all their data. So it gives us a level of resilience that actually we couldn't get by sticking within a single university. And we've explicitly designed the system to enable the addition of other universities and other nodes. And that's pretty important to us because we think that this is actually a really great way to present infrastructure out to researchers. It's a way that most researchers can understand, certainly in the biological sciences. And so the idea being that as other universities start to invest in updating their infrastructure or putting in cloud infrastructure of their own, we're designing a system that's built with the explicit expectation that we can then add in extra sites to create an even larger system that spans multiple universities. And I guess the key thing here is that this project is academic-led. So I'm a researcher. The leads at all the other sites are researchers. And we're focused on the community that we're delivering to. We're part of the Microbial Bioinformatics community by the simple fact of the way that we do our research and the people that we know. But this isn't something that's been done in competition with local HPC. It's not something that's been done in competition with local IT. It's been collaborative throughout. So we've got what I think is quite a unique and very collaborative approach, not just within institutions. So, I mean, I work very closely with the people within Cardiff, but between institutions as well. And it's really nice to see that actually the team works really well. And so, it's a real advert actually for how to do these sort of things. It's difficult to get over that initial step of, well, how do you get to know these people? How do you build that network? But once you've got the network of people, around a clear project, and especially an exciting project which really is pushing the boundaries of what's technically possible, then suddenly everyone clicks into gear and it seems to be going all right. So we have 4 sites. We're running OpenStack at the moment. Hardware is procured in a 2-stage process. IBM OCF provided compute, and Dell Red Hat providing the object storage. The networks are all Brocade, and we're defining a reference architecture to enable other sites to trivially be added. So we've got various elements within the system which are critical. So the cross-site replication is through this Ceph, the object storage system, and so obviously defining that, we're finding that there are more gotchas than you might expect when actually setting up, actually even down to picking nodes and around things like the RAID cards and the host bus adapters on the Ceph nodes. And so there's actually a lot of knowledge that we're accruing here about how you set up these systems, which will be of wider use to the UK academic community. And I mean, things like the Brocade system. So internally we have the high performance. So IBM GPFS provides our local storage, and that's all connected over Mellanox InfiniBand. And then we have a failover onto a Brocade BDX fabric. So our OpenStack network and the network which then connects to the outside world is a Brocade 10 gigabit BDX fabric. And then at the gateway to the system, we have a pair of Brocade Viatas sitting there, which are an interesting thing again, which because it's a relatively new product, it has a lot of essentially very useful functionality, and so we feel we're really pushing the boundaries on how they're being used as well to do what we want to do. So that's some of the IBM nodes. First sites, we have 2 routers / firewalls, and each of those is capable of routing about 80 gigabit. So they're pretty hefty. We have a hefty spec, and the Viatas perform very well. Each site has 3 OpenStack controllers. We have 21 64-core 512 gigabyte RAM nodes per site. We have 3 192-core 3 terabyte RAM nodes per site. About 500 T of GPFS locally, and they're connected over InfiniBand but with 10 gigabit failover. And then we've got as I say, between 2 and 3 petabytes of Ceph as we've currently got it configured, but we're currently scaling that as we test. And that gives us quite a substantial individual system with a whole host of technical challenges. That's what we've spent the last few months working through. So we've got all sorts of challenges. Federated access. At the moment, OpenStack implements a shibboleth module via Apache for authentication. Getting that to work is a whole category of fun that we're currently sorting out. And actually, the federated access, how that then interacts with OpenStack for generation of tokens and how it works across site for tracking things like the virtual machines is something that we're currently working on, because obviously when you've got universities that are separated by 50, 60, 70, 80, 90 miles, latency becomes an issue. So actually working out how we can implement that is something that's ongoing. We've got what we think is a working system, but it's something that we're still testing. Our storage configuration, we've had various interesting issues around the GPFS. The GPFS is meant for running the virtual machines and also for storing sort of scratch files for user processing. And basically, we've been playing with the block size to try and ensure that we get the performance that we want out of the GPFS system, and it's by no means as trivial as you might have hoped. So actually choosing the block size is critical for the VM performance, and you take a huge performance hit if you get that block size wrong. The networking itself, so the VDXs were the easiest part of the system to put in. We have so at Cardiff we have 3 VDXs connected together, and they create a single VDX fabric. So they're a single, and I think they're in logical chassis modes. They present effectively as a single switch, and they took very little time to configure. They went in, they were connected up, and they worked from day one. So they are, I think, the only time I've ever seen what the vendor said was actually true, and so our experience has been very good with the VDXs. But actually, then you move on to the complexities of site-to-site connections and site-to-site VPNs through the VIATAs, and that's another area that we're working on, what the best way of doing that is. Obviously, we have this issue of latency between sites, working around that, and we have solutions for that, but they're currently being tested. And finally, we have this issue that because a lot of biology is workloads that haven't really been very well defined or aren't well defined now. So a lot of bioinformatics is about responding to advances in technology to use that technology in the best possible way to answer your research questions. So we are explicitly having to design a system for unknown use cases. And so we knew, based on the profile of our workloads, that we'd need relatively large amounts of RAM. But then you start to run into issues about people coming with a workload that maybe needs more CPU cores. And so one of our challenges in the first place was designing a system explicitly for those unknown use cases. And now it's about, well, how do we use the infrastructure in the best way to mean that it can be used by as many people as possible. So I mentioned the block size of GPFS as one of our implementation issues. We're still working on VPN tunnels between sites. So there was, until recently, there's been an issue with the VIATA software itself that's limited our ability to create the point-to-point VPN tunnels that we want to securely move the data. I mean, it might be that actually this ceases to be an issue with current initiatives within JANET around providing a secure network for moving data around. But at the moment, that's an issue for us. All sorts of difficulties around high availability. So in the Genome version of OpenStack, high availability is a lot flakier than it should be. There's a lot of complexity around how you set it up. It's not something that's inbuilt within OpenStack. We know that within Kilo, that's supposed to have been addressed, and there's a new mechanism for high availability implemented there. So we'll be seeing, we just upgraded the Warwick and the Swansea sites to Kilo, so we'll be actually testing and seeing if it is actually better. And then we find that both OpenStack and Ceph are extremely complex pieces of kit, and there's a lot of gotchas in there that aren't really published well online or are very difficult to find online. And so this is where the teamwork comes in. Is we've got 4 sites worth of HPC staff who are all extremely experienced in all manner of things, from networking through to optimizing performance. And actually that gives us a really useful pool of knowledge, so that when we run into an issue, chances are someone on one site would have seen something similar in the past and will be able to work through it. So generally our performance is extremely good. I've actually just spent the summer benchmarking climb against various other services. I'm not actually putting them up here, but it performs. So Raven is our HPC system at Cardiff, and I've got a couple of things in here. So when you've got the incorrect block size, you see a lot of dropouts in performance. These are various bioinformatics workloads. But when you've got the correct block size, almost in every case we're performing better than the local HPC system. And so you'd sort of expect that because we've got processors that are a generation newer than the HPC system. But it is interesting because you hear about various quotes about the performance hit you take from virtualization. All these benchmarks are run within VMs, other than the HPC one. And so actually the performance is a lot better than we'd expected. And funnily enough, the performance hit from virtualization doesn't seem to be too big. There's only a couple of workloads where virtualization really impinges on the performance. So our performance is pretty good. It's pretty consistent across workloads, which is pleasing to us. But we really see how critical the configuration is. So the correct block size is one issue. Another issue that we've got is that within the virtual machines running on the high memory machines and the 3 terabyte RAM machines, we see a real drop in performance, and we're currently investigating the reason for that. We think it's hypervisor related, but we're not sure quite where. We think it's possibly because these boxes are IBM 8-core boxes which have a backplane in the middle that connects up, what they call it, a mid-plane, that connects up effectively 2 4-socket servers. And we think that it's something to do with the scheduling across that, which is causing by the operating system and the hypervisor, which is causing the issue. But we're not 100% certain where the problem lies yet, as that's something we're currently working on as well. So it's a really weird issue with large memory machines. It's not really been reported anywhere else, but then I don't know how many other people are actually running 3 terabyte RAM machines in sort of production environments. Certainly not who are going to publish results about where they have performance issues. So that's one that we're currently investigating. So in terms of our timescales, we got the award letter in April 2014. The procurement process really kicked into gear in August last year when all the contracts were finalised between the universities. And everything was in place and acceptance testing completed by March this year. And we've got, and as I say, over the summer we now have 2 sites in full production. We've got about 45 users from across the UK microbial bioinformatics community who are currently what we call our early access adopters. And so we're spending the next few months now just ironing out any of the problems that we've got within the service in order that then we can hopefully have a launch towards the tail end of the year. We're also now starting to move on to the slightly more difficult parts, which are around developing new interfaces and new ways of actually accessing the system, which we'll need for the majority of our users. That's the summary there. So we have access for registered users to a core OpenStack system which is available online now, and you log in and there's a self-service interface, and you can choose to spin up a virtual machine of whatever size you want, as long as it's within your quota, and you get a view like this as part of your quota, which tells you what's available. At the moment, the Birmingham site is 100% utilised, so we have virtual machines which are currently using all capacity on that system, so it's already being used for research. We're currently, as I say, playing with the configuration on Ceph. It's up and running on 2 of the sites at the moment, and we're just testing things like replication between sites and connectivity into the VMs to make sure that everything's functioning as it should be. And we're currently configuring that multi-site connectivity and the authentication for that. So we've got lots of VMs up. You can see that there are certain groups, one particular group in Warwick, who are developing a database that's for sharing data on enteric pathogens, who have been heavily utilising it. So, I mean, I think I took this one earlier in the year, but thousands of CPU hours and terabytes of disk gigabyte hours. So it's being used, and it's going pretty well. And we're starting to be able to do things that we couldn't do on an HPC system. So this is just a screenshot of the top that shows a 3 terabyte RAM machine being pretty much 100% utilised by by a software assembly package called Ray, which assembles metagenomics sequences. So the issue that we start to run into with smaller memory machines is that there are some workloads that we're physically unable to do with the current software that's available. And the only way to deal with that actually is to get more RAM. And there aren't many universities that have 3 terabyte RAM machines, and that really opens up new possibilities in terms of the sort of science that we can do. So it's a pretty complex system, got a lot of challenges. We started off with a pretty clear vision and a pretty clear idea of what the community was that we were targeting in terms of how we were building it. I guess part of the complexity is that it's not just built on a single site, but it's built across 4 universities, so we run into issues around different policies and different perspectives from different universities, and also managing a large, complex project like that across 4 universities, 4 sets of HPC teams, all with different draws on their time, is not trivial, but actually I think it's worked pretty well. And absolutely key to this is close collaboration between the technical team and then allying that with a really clear strategic vision for the project as a whole. So that's just a very quick summary of what we're trying to do. So a one-stop shop for microbial bioinformatics is our tagline, and that's the client consortium. And I thank you for your attention. So I have a suspicion that if the system is highly utilised and is used by a lot of researchers to publish a lot of interesting science, then in 4 years' time, when the project runs out, we'll have a case to go to the MRC to ask for more money. But what we're not sure about yet, and so I should go back a step and say, we're researchers and we're not necessarily interested in running a cloud for the rest of time. And we're partly expecting that in terms of the general compute that you might want to do, in 5 years' time or 4 years' time, it's probably going to be cheaper for a lot of this stuff to be pushed down to something like Amazon or to one of the other public clouds. And in that situation, I guess our focus changes from necessarily providing a cloud to actually providing a brokerage service where we can make the software and the data that we store available elsewhere on other public clouds. And so I guess our long-term plan is that we're waiting to see exactly what happens in terms of the cloud space. And I guess my expectation is that probably in 5 years' time, what we'll be doing is not asking the MRC for another £3.6 million to replace this. We'll be asking something for a substantially smaller amount to provide a replacement for the storage required to actually share the data and the software and to broker that out into the cloud. And so that's my sort of expectation. But it's a difficult one to know because you can't predict how prices are going to change. And there are various excrucible things at the moment. Like one of our advantages that because this is a system hosted within UK universities, it makes things like terms and conditions and policy much easier in a sense for UK researchers than say putting your data out to Amazon and agreeing to their terms and conditions, or putting it out to Google and agreeing to their terms and conditions, and possibly falling afoul of European legislation or whatever. Because we're running this locally, we can get around that. And so there's a series of things that have to be covered off, I think, in the next few years. But realistically, you would sort of expect by 2020 that the public cloud services will be cheap enough, or will hopefully be cheap enough for the majority of what we do, that we would look to be making our software and our data available in those rather than necessarily hosting a service ourselves. If that makes sense. terms of what?
In terms of if there is a public data output from
Yeah.
so what you do, what it is you set it up
Yep.
to, would you change your course of interaction? Or I suppose they're given the funding,
Yep.
and you set in your direction.
Well, I guess. I guess there's two sides to that. So the first is that if there's always a cost, right? So the difference here is that we can provide this service for free to UK academic users. And so for a lot of microbiologists, this removes the need for you to have your own local servers or whatever. So in terms of servicing the community, the chances are we probably carry on doing that because we sort of feel like this kind of infrastructure should be available for free to the UK microbial community. And so, again, until, I guess, the funding regimen changes. So if there was a public cloud that researchers in the UK could get unfettered access to, then yeah, we might reevaluate exactly what our focus was. But if it was the case that it's still going to require you paying for access to that cloud, and that cloud doesn't provide access to, say, large memory machines, so the thing is, our VM flavors will scale all the way up to 512 gigabytes of RAM, which is larger than anyone else can do at the moment. And for some workloads, so we've got some large Bayesian model-based approaches which require extremely large amounts of RAM. And so there are a whole host of workloads that actually this is quite good for. But yeah, our expectation is that the need for a publicly funded service like this is going to decline with time. But at the moment, it's quite I think it's quite a well-positioned thing in terms of what we're going for. So have you're trying Ceph, are you?
Yeah.
That's with Inktank and Red Hat, is it? Okay. So ours is fully supported by Red Hat. So we have consultancy days. We have direct contact with their developers. So actually that's been quite good. We still have a few issues around the hardware itself. So we've had a couple of issues with disk performance, which we're trying to nail down at the moment. But again, I have to say, both Dell and Red Hat have been very responsive to what to our issues. The other thing I should say, the other bit that we've done, so Climb includes money for some system administration, and one of our first priorities was to hire Marius, who has run Ceph in a production environment previously for companies, and so Marius is our Ceph lead. And he's interacted with the developers at Inktank for some time. So actually, my other suggestion is, if you want to go down the route of having something like Ceph, it's like anything else in IT, make sure you've got an expert in your camp who can do it. And Marius is the one who's pretty much in charge of rolling Ceph out and making it work. It was half and half. So the way that we bought some hardware at Birmingham through their existing HPC framework early on to do some development work on, and then when the system went in, they fed into our experiences from doing the development work to inform how they set it up. So the recent upgrade we just had from Juno to Kilo was entirely done by OCF. So they came in, they upgraded it. And that was it. So we haven't had to do anything there. So we've actually had a really good exchange. Again, I think the thing is that this is an interesting project from a vendor's perspective as well. So we've had a lot of buy-in and a lot of interest from Dell and Red Hat and from OCF and Lenovo. And I think that really benefits the project as well. And so we've got bits and pieces, but actually we've had pretty good, connectivity with them. Obviously accounting for things like holidays over the summer and that sort of thing.